Yeah. עוד פעם, איך עושים הפוינטר הזה? כן. אוקיי. אז אנחנו... Okay, I will switch to English. We continue with our course, Pharmacology and Physiology of Neuropsychiatric Disorders. Before the next lecture, I will make few organizational notes. First of all, with your kind permission, we already discussed it, I would make a small change in the syllabus. Today we continue as planned interaction between monoamines. No, today we continue as planned autoregulation of monoaminergic neurotransmission. It will be the topic of today. And tomorrow, next week, next Tuesday, it will be again a regular in-person lecture about interaction between different neurotransmitters. And then I will go back to several days back to Slovakia. And from Slovakia, I will deliver to you, I will organize interactive visit to my laboratory. And we will choose some interesting experiment. We will show you it again via Zoom with all explanations. It will be in the same hours like we do now. By the way, if somebody interested even to join me physically, it's also possible. Well, I don't have funds to support it, but you can find now flight tickets for less than 1,000 Israeli shkalim. And if somebody, you can go to Vienna or to Budapest. And if somebody of you choose it, contact me, and I may help to find an airport transfer, free airport pickup, and maybe free accommodation in the guest room of our institute. But I believe it's unlikely that somebody will go. You are busy with other issues. Then, because I see we go faster than I expected, the next lecture after it will be on 16th of July. It will be in person again, but we will merge two topics. We will speak about histamine and adenosine instead of to make each of one as a separate topic, as a separate class. And then we continue as planned. Another organizational issue. I have my guest office here in Sharet building, room 105 Aleph. I plan to be in this room Sundays and Tuesdays from 11 to 1 o'clock. You're very welcome to come, but better today I leave straight after the class. Not today, but other days, and please feel very free to contact me. I would like I already to tell you about final works, Avodat Gmar, what I expect in this course, what I would like. As you know, it will be a final work. It will not be exam. And in final work, but I'm a little bit skeptical about productivity of the seminar works when somebody choose one paper and present this paper. I don't really feel big toilet, big output of that. Instead of it, I would like you to feel when you will do. Don't worry about the find the marks. I'm usually very good with the marks and with the grades. But I would like you, when we'll come to final work, I would like you to feel researchers. For a while, you will feel for a while as a head of the lab, as a PI, principal investigator. You will choose one or three papers with some idea that say that, for example, some kind of molecule, let's say melatonin, interacting with serotonin. You will propose mechanisms how it may work, and you will suggest how you will check it experimentally. It will be mini research proposals. Of course, fully theoretical. It's up to you. You prefer to work in pairs or personnel? Yeah, if I think university rules, they okay. It's up to lecture, up to teacher, yes. then for me it's okay. Optional. I will get extra bonus for somebody who will choose to present. You don't need to submit written before presentation. You can make presentation and then submit written part later. אתם יכולים, אתם יכולות גם לעשות את זה חלק הכתוב, להגיש יותר מאוחר. זאת אומרת, ההצגות זה נעשה בזמן הקורס. בשיעורים האחרונים אנחנו נגיש אולי 10-15 דקות בכל להצגות שלכם.
Would it be possible to upload the instructions to the envelope?
I will. I still learning how to use it. I see you need to have. I see you need to have to degree how to use this court management. Okay, and let's go to today's topic finally. Today we will speak about autoregulation of monoaminergic, mostly serotonergic neurotransmission, and the role of this autoregulation in the response to antidepressant drugs. We already started last time to touch this topic, and we asked a big question. On the left, you see microdialysis study, neurochemical study, which show how fast we have increase in the brain serotonergic levels after injection of escitalopram. You see, if rat get injected with escitalopram, within 20 minutes you have already 400% increase in serotonergic levels. But what goes on in clinics? This is a clinical study with exactly the same drug as escitalopram. And you see that you start to see clinical effects, some relief in the symptoms only after 2 weeks. After 1 week, it's absolutely no difference between placebo and escitalopram. And our question was, what's going on? Why drug, which is so fast acting from the pharmacological point of view, why it takes so long to obtain clinical effects? And one of the first answer why it can be because the autoregulatory mechanisms of serotonergic neurotransmission. This is serotonergic nerve terminal, which we saw before. Serotonin get released and get reabsorbed back to the presynaptic cell. And if we block serotonergic transporter, we get more serotonin in the synaptic space. But at the same time, we get more activation of autoreceptors. Autoreceptors, which they expressed on the presynaptic neuron, and they negatively regulate activity of this presynaptic neuron. And one of the questions maybe this autoregulatory mechanism contributes to this delayed response to antidepressants. What makes situation a little bit more difficult that we have two types of, in case with serotonin, we have two types of autoregulatory negative feedback autoreceptors. It's 5-HT1A autoreceptor. It's shown here in the orange, so it's red color, and in the blue you see 5-HT1B autoreceptor. 5-HT1A autoreceptor expressed on the cell bodies of the neurons. Where cell bodies located, you remember? Cell bodies. Who remember where cell body? Exactly, yes. And 5-HT1B receptor is expressed on nerve terminals. It means that it can be found everywhere in the brain. Somebody can tell why they shown by 7 lines? Yes, because it's all G protein couplet receptors, they have 7 transmembranal domains. And they both negatively regulate activation of these receptors. Both of them results in the reducing serotonergic release in nerve terminal. but mechanisms are different. Mechanism of 5-HT1A autoreceptor involve downregulation of the firing activity of the neurons. When we activate 5-HT1A, we have neuron become less active. But when we activate 5-HT1B receptor, we have decrease in the neurotransmitter release, but it's happened via mechanism which does not involve firing activity of the neuron. And you can already tell me, yes, and what's probably going on with antidepressant, delayed antidepressant response. This is a basal condition. This is a neuron, schematic representative of serotonergic neuron. Here you see it's a 5-HT1A autoreceptor. On nerve terminal, we see 5-HT1B autoreceptor. And then we give escitalopram. We get increase in, first of all, we get increase in 5-HT in raphe nucleus, in cell bodies area. And we have activation of 5-HT1A autoreceptors, but also of 5-HT1B autoreceptors. Firing rate going down, and really, and we don't need even we have inhibition of reuptake. We have more serotonin in raphe nucleus, but we don't get more serotonin in the nerve terminal area. What is more important to us? And after 2 weeks and longer, this receptor desensitize 5-HT1A and 5-HT1B. We will see later what does it mean, desensitization, and how it happens. After desensitization, we have restoration of the original firing activity of the neuron. And when we have reuptake inhibition with the restoration of the normal firing rate, we get finally antidepressant risk. We get more serotonergic transmission, more serotonin released in the nerve terminals. How we can check it experimentally, the reactivity of 5-HT and 5-HT1A and 5-HT1A receptors. Because 5-HT1A receptor mechanism of action involves the firing activity, the easiest way to check this will be by in vivo electrophysiology. It's a method, it's a key method in my lab. I hope to, in 2 weeks, I hope to show you in vivo electrophysiology experiment. And we insert electrode to the dorsal raphe nucleus, and we detect spontaneously active serotonergic neurons, as you see here. And during the recording from one neuron, we inject escitalopram. And here, for some reason, I cannot play this movie. But you saw it last time, this movie. And you can play it when you when I will upload this presentation to the course website. You see, this is what's happened when we give escitalopram serotonin reuptake inhibitor acutely. After its acute administration, firing rate of the neuron go down till it's totally blocked, till it's totally shut down. And when we give this compound, weigh 135, we have restoration of the firing rate of the neurons. And who can tell me what this compound is? It's this compound is antagonist of 5-HT1A autoreceptors. It means that indeed escitalopram inhibits serotonergic firing activity, and indeed this is mediated via 5-HT1A autoreceptors. After 2 days of treatment, after subcross, it's acute administration, immediate, with an immediate effect. Here you see effect of 2 days treatment. Rats were treated sustainedly, repeatedly for 2 days. Still, we have potent inhibition of the neuron for 5-HT firing activity. But after 14 days, this inhibition disappears. It means that indeed the process after 2 weeks happens, we get the desensitization of autoreceptors. 5-HT1B autoreceptors, they do not change firing activity of the neuron. So how we can check their reactivity? We can use microdialysis, which we already reminded this method. We insert probe with a semi-permeable membrane again to the dorsal nucleus, and we can check change in serotonergic levels. And this is, by the way, this was my first published study when I was I was only my first year master student. And till now, I think it's one of my best studies, my math-based papers. Here we see rats which were treated for 2 weeks with fluoxetine. Citalopram was still not existed. Fluoxetine was the most used antidepressant at this time. It's a Prozac. And with white circles, it's rats which treated with just with a saline, with vehicle. And black dots, the rats which were treated with, no, other way around, fluoxetine, the white dots and black dots, saline. And then we implanted to these rats probes and we injected GR127935, which is antagonist of 5-HT1B autoreceptors. And you see which rats with pre-treated with saline, we get increase in serotonin, because 5-HT1B autoreceptor negatively regulates release of serotonin. If we block this receptor by antagonist, we get increase in serotonin. But rats which get treated with fluoxetine, they don't have this increase. It means this receptor also dies after 2 weeks. And I would like to show one interesting situation when electrophysiological investigations and microdialysis assessments they confirm each another. You remember this slide, yeah? It's like in the joke that police investigator ask, "Do you recognize this gun?" "Yes, I recognize. You showed me already 10 times it." So it's exactly here. I show you. I repeating showing this picture from the first class. What we saw that escitalopram is a racemic compound which contains 50% of escitalopram and 50% of arcitalopram. We saw that arcitalopram does not has had any effect on serotonin release. It does not effectively block serotonin transporter. But escitalopram, racemic citalopram, even it consists same amount of escitalopram, is much less effect than pure escitalopram, which means that arcitalopram not only blocking effectively, it's also preventing escitalopram from blocking this receptor. And Mustafa El-Mansari, I was very happy to work with him in Canada. He's lab manager of my postdoctoral supervisor, and he was my actual direct supervisor during my postdoc in Canada. And he confirmed the same paradigm electrophysiologically. You see, he applied escitalopram during the recording from a serotonergic neuron, and neuron gets shut down, exactly like in my study, which was done later. When they was injected, it came back. You see, when escitalopram does not have the same effect, even when he applied 500 microgram per kilogram citalopram, which supposed to have 250, two times higher concentration of citalopram, the effect was less. And arcitalopram blocks. If you pre-treat rat, first give arcitalopram, then escitalopram cannot work anymore with the same effectiveness. So,
the same target?
yes, they have 2 binding sites, and if arcitalopram occupy one binding site, escitalopram doesn't work anymore effectively from the other binding sites. What does it mean? I promised you to talk today. What does it mean? Desensitization of the receptors and how it's happened. You remember that there are G protein couplet receptors. The activation alpha subunit inhibits adenylyl cyclase. But what is very interesting, beta gamma subunit activates phospholipase C and another kinase, which is AC2. And this activation changes activity in activation of adenyl of PLC and AC2 leads to phosphorylation of this protein of this autoreceptor 551N or 551B autoreceptor. After it gets phosphorylated, it's already start to be less effective, less its affinity to the substrate, to serotonin, is going down. It's like first step in desensitization. It's still function desensitization when the expression of when the density of the receptor will be the same. Second step, this phosphorylated receptor recognized by some kind of mechanism, which probably involve beta-arrestin, and this receptor go internationalized. It go inside with a piece of membrane, it go inside to the cell. And later future of this international receptor depends on the level of phosphorylation, again on the level of activity of the specific protein kinases. It can be recycled back to the cell membrane, or is going to be destroyed by lysosomes. I will not go deep to this point in my course, but it can be, if somebody would like, for example, to choose this point, molecular mechanisms of desensitization of the receptors and how we can intervene with this problem, it can be one of the topics. So let's now from molecular point go back to pharmacology. And what can we tell if the problem in autoreceptors, if the autoreceptors are bad guys and we have delay in the effect of antidepressants because of the activation of the autoreceptor, let's block them and we will get fast antidepressant response. What do you think about it?
Wait for a toxic effect.
Like good way for
Toxic effect. Guessing that the autoreceptor modulate activity of the neuron.
yes.
So if stop the modulation, there can be overactivity.
Yes, but we need because depression is probably depression a result from decreased serotonergic neurotransmission. We need to overactivate it. First of all, one of the treatments for depression, for severe depression, even no drugs working, it's electroconvulsive therapy. When we get extremely severe activation of the neurons. So what's the problem? Let's do it. And to be honest, it was done. And it's still used in the clinic, in the clinics. There are two, not very much they used anymore, but still used. There are two drugs, Pindolol. It's it's a synthetic compound, and it blocks 5-HT1A autoreceptor. They cannot be used for some reason. I don't know exactly why, but they not used in clinic, but Pindolol is used. Maybe you're right. Pindolol is a partial agonist. It's antagonist, but less effective as antagonist. Maybe you're right. Maybe they is not used in clinics because with the overactivation we can get some toxic effects, serotonergic syndrome. And another compound which used to modulate autoreceptors, it's a block 5-HT1B autoreceptor. It's T3. Somebody heard about T3? No, you never heard about it. Exactly, it's a tyrosine, it's one of the main tyrosine hormones. Thyroid gland, it's pretty thyroid system, pretty complicated system. It has 3 levels. We have TRH, which released from hypothalamus. It's activates TSH, and TSH activates TSH release from hypophysis, from pituitary, and it's activates T3 and T4 hormones. And we know that thyroid dysfunction very, very linked with depression. About about half of depressed patients, they have some kind of thyroidal signaling dysfunction. And people with hyperfunction of thyroid glands, they very commonly also depressed. So these two systems, this thyroid system very connected with depression. And one that was very big interest in thyroid system and thyroid hormones as a treatment in depression. Unfortunately, it went down, this interest. And this is work, Dr. Michael Newman, he was my supervisor during my master's studies. I considered him my primary teacher, my most significant teacher. Unfortunately, he passed away pretty young and from the brain cancer, but he continued to work to the very end of his life. And it was his last paper, which by coincidence I was very glad I was guest editor in the journal, and I was last to accept his last paper. And his last paper was exactly about this topic, about interaction, about role of T3 in autoregulation of serotonin release and antidepressant response. I also recommend to choose it to one of them, because I don't go deep to this topic. You can choose it. Somebody who wish can choose this topic about T3 and monoaminergic interactions. But it's not so simple as I told you. We told now, okay, 5-HT1A, 5C1B autoreceptor bad guys, let's block them. But let's check pharmacological profile of vortioxetine. Vortioxetine is a very large generic. I think is the youngest antidepressants developed by Lundbeck. It's very effective drug, very good. It's a serotonin reuptake inhibitor, and put attention, it's agonist of 5-HT1A receptor. So come on, if we need to block it in order to get more serotonergic transmission, why this drugs is activating 5-HT1A receptor? Somebody can tell me, tell me how it can be solved. No, well, 5-HT1B is a partial agonist of 5-HTB, so it's okay. Partial agonism more or less like antagonist. But it's agonist of it's activate 5-HT1A. If you activate 5-HT1A, you shut down the neuron. The answer is that this is the same receptor, 5-HT1A receptor. It's acts as a autoreceptor on presynaptic neuron, but it also acts as a heteroreceptor on postsynaptic neuron. So if you block it here and here, and you have less transmission. It was one of the so-called holy grails of drug developments to develop to find some kind of antagonist which will act only on presynaptic receptors. It was a lot of power, energy was invested, and money was invested in this direction. It was some hints, it was from time to time a report as a little bit pharmacologically different this receptor, but nothing worked out. It looks like from the outside, from the extracellular side, they're absolutely identical, presynaptic and postsynaptic 5-HT1A receptor. They're a little bit different from the intracellular side, from signal transduction mechanism. So what can we? But there is one question: if presynaptic receptor, if they so similar, why presynaptic receptor desensitize, and why postsynaptic receptor do not desensitize? What do you think? Exactly, it's you're very close, very hot. And answer, because they have different signal transduction mechanisms. And signal transduction mechanism is depending on other type of adenylactic and protein kinases which are expressed in this specific neuron. We see that in presynaptic neurons, in 5HT neurons, it's activate only PLC and adenylacticlase and inhibits adenylyl cyclase, but in postsynaptic neurons, it activates plenty of other signal transduction mechanisms. Again, if somebody wants to go, I'm not molecular biologist. All molecular topics, I only mention them, I don't go deep, but somebody can choose it, different signal transduction and how we can use different signal transduction in order to create more specific drugs, for example, which will act mostly pre or postsynaptic. Somebody can choose it as a work. Okay, but still, what can we do to target presynaptic receptors specifically? If 5HT1A receptor postsynaptic and presynaptic, if from the outer side, from outside of the cell, they similar, what would you suggest how you can target them? How you can target, what you can do that specific drug, specific blocker will arrive only to presynaptic receptor and not to postsynaptic receptor. But yes, do you have any idea how it can be done? Very good. It's a good answer, and I would divide answer to two. First part is scientific fiction. What can be in future done to do it? One, it's so-called, it's very hot topic in drug development now. It's called targeted delivery. It's how to do, because each drug, it's not necessarily neuroscience. For example, in cancer treatment, it's much more actual, because all chemotherapy, all chemotherapy drugs are very toxic. So how to do it, for example, to deliver them exactly to the tumor. And in neuroscience, what can we do? Let's say this to take 5HT1A antagonist and deliver it to the roughly nucleus and not to other part of the brain. One part, we can encapsulate them in nanoparticles. These nanoparticles can have on the, for example, Plex, it's one of the most commonly used now nanoparticles for the intraCNS delivery. We can attach to this nanoparticle specific proteins. These proteins can be, for example, antibodies to the transport proteins which transport anterograde and retrograde transport of synaptic bezels. And by this way, we can theoretically reach specific area of the brain. One of the projects that is running in my lab, but we try to deliver BDNF coding mRNA straight to the brain using this targeted delivery. Another targeted delivery mechanism can be, we can use physical electromagnetic power. We can use, it's also used for the cancer therapy. We can take compound which we want to deliver to specific area, let's say to the DRN. We can make encapsulated into the magnetic nanoparticles, into the nanoparticles that have ferromagnetic components. And by external magnetic field, we can force them to come to run to the specific area of the brain. It was a topic I tried to submit to one European grant, but it was rejected. Yes. Because presynaptic neurons, they mostly locate. If you speak about 5-HT1B autoreceptor, it will be tricky. But 5-HT1A autoreceptor, you can 5-HT1A receptor, you can easily differentiate between presynaptic and postsynaptic by brain location. In the rough nucleus, they only because they express on cell bodies. In the rough nucleus, they only presynaptic. Because they not express on the nerve terminals, in the hippocampus, they will be only postsynaptic. So if you let's say encapsulate this drug to magnetic nanoparticles and force it to the rough nucleus, it will block theoretically only presynaptic receptor, not postsynaptic receptors. This was fantasy. It was all fantasy. But the problem in reality, we need to treat patients now. And all these targeted delivery methods, they will be developed, or they will not be developed, but even if they will be developed, it will be in the not so close future. So what can we do to treat patient now to target specifically receptors, presynaptic receptors, to make presynaptic receptors, to make them less active and not postsynaptic receptors. There are many. In this course, we are mostly speaking about pharmacotherapy, but pharmacotherapy is only one of the methods used in the treatment of the psychiatric disorders. Another method is psychotherapy. About psychotherapy, I don't know much about it, and we will not talk about it. Probably some of you studied psychology, so I believe some people here know it much better than me. If somebody wants, by the way, it will be very ambitious to show neuronal mechanism of psychotherapy. If somebody wants to do it, I will be very glad to see such a work. We have physiotherapy. Physiotherapy is a treatment with physical methods. There are two common physiotherapies used in psychiatry. It's electroconvulsive therapy, ECT, which I already reminded, which is today ECT might become much more patient-friendly, not aggressive like it was in the past, but still it's a useless last resort treatment because it has side effect and people normally scary about this treatment. Another treatment, another physiotherapy is a transcranial magnetic stimulation. We slightly reminded in the previous slide. It's we can activate by external electromagnetic field specific area of the brain. Finally, there's psychosurgery. If you heard something that psychosurgery is prohibited, it's not true. Some forms like lobotomy, it was one of the first psychosurgeries. It indeed prohibited in most of the countries, but some psychosurgery is still used. Again, as a last resort treatment. For example, with a very severe obsessive compulsive disorder, which is not managed by drugs, resection of cingulate gyrus is used as a treatment, and combination of physiotherapy and psychosurgery. It's deep stimulation. It's a vagus nerve stimulation or deep brain stimulation. But we forget there is one physiotherapy that we forget, and somebody can tell me which physiotherapy we forget. I forget to mention. Some ideas. Now, should I click to the next slide? It's a physical exercise. Physical exercise actually has fantastic psychotropic effect. It's got fantastic antidepressant, anxiolytic effect. And maybe even we have such a prevalence of depression and affective disorders because of the sedentary lifestyle. And yes, if you want to see like clinical studies about effectiveness of physical exercise, there is a wonderful review written by Kvam in 2016. He made meta-analysis, and for example, at least 4 studies show that physical exercise, voluntary physical exercise, has a strong antidepressant effect if you compare it with no other treatment. What is good with the physical exercise is that it's not specific to humans. Animals, they like to exercise very much too. Who of you is a pet owner? Only you? Dog, cat? Dog. I think dogs they like to exercise more. Cats is very usually cats is very sedentary animal, but cats also run and exercise, and cats they also exercise. Rats, if you attach running wheel to the cage, rats very like to use it. Not as a hamsters or squirrels, but rats they like. Furthermore, we have big question if our standard keeping of rats actually maybe our standard way how we keep rats and mice in the lab is not so good because we prevent them from necessary exercise. It estimates that regular Rattus norvegicus, which live in nature, no, not exactly in the nature, which live in the cellar and our cellars, they usually spend around 10 kilometers moving in the day. And this is a study of Hen, published in 2016. He found that indeed, like in humans, wheel running in rats have pretty strong antidepressant-like effect. He made rats to run he allowed rats to run in the wheel for several weeks. Then the rats underwent swim test. You remember swim test? And he found that rats have significantly decreased immobility time and slightly increased swimming and climbing times, which means that it's hard to say. Or rats which allow, or well running has antidepressant-like effect, or running rats say normal, and rats which are prevented from exercise they are depressed. It also can be like this. Yes.
Spend more time doing planning than
Exactly. This is definitely can be criticism to the study. And for example, more like I will say that elevator plus mate would be more. But in EPM, as far as I know, it was no effect were not found. But even you fit why you should swim, because if rats can rats is not drowning even if he is not swimming. But definitely, I agree with you. This is a point of this is limitation of the study. It's another study from another group of Greenwood, 2003. I brought the study only to make you introduce to you very, very important research techniques used in neuroscience. And this techniques is C4 immunoreactivity. Somebody heard about CFOS? Yes. So I don't need it. I will explain briefly. CFOS is actually a proto-oncogen. It's a proto-oncogen of the oncogen VFOS. Neuroscience researchers use as a marker of neuronal activity. As much neuron is active, as more CFOS is expressed in this neuron. It means if we found such increase in the CFOS expression in the dorsal raphe nucleus, it means that yes, but we cannot know which type of neurons. If it's serotonergic neurons or no, actually he did check it. He checked double expression of CFOS and tryptophan deoxylase to confirm that the serotonergic neurons. And we decided to make, it was one my first study that I did when I moved to Canada, when as a PI, as a head of the lab, when I was head of the lab and only member of the lab. I was a PI and the postdoc and the PhD student in Belarus. At that time, it was my first work that I did in Canada. It was started 10 years ago and was published 7 years ago. We made it was technical model well developed in the laboratory of my primary collaborator Daniela Yezhova. We allowed rats to run for 3 weeks, and what you see as far as more than a rat run, they start to run for more and more distance, and they spend more and more time in running till it's come to any kind of plateau. But there is not much difference in the average speed of the running. Yes, some rats they spend up to 10 kilometers. They're very different, by the way. They probably, in the terms of sedentarity and liking of exercise, rats, they're very different like humans. For example, I feel very bad that I'm a very sedentary person by nature. I need to force myself to any exercise. And some people, they really enjoy very much exercise. Same we see with the rats. By the way, some rats, they spend only 3-4 kilometers, but some up to 12 kilometers running.
This changed by age?
It's a very good question. We never checked it. Probably yes. We use relatively young rats. And it's differently different between strains of the rats. For example, I usually use in my lab Vista rats because they're available in Slovakia. But for this experiment, I had to use straight dolly because Vista, they're very sedentary. They don't like to run. They like little. And from other side, Vista, they must less anxious. So, yes. Yes. It's a very good question. Hard me to imagine non-aerobic model in rodents. In humans, I don't know. It's a good question. Need to be checked. Probably in humans, it might be also non-air. It's how is called non-aerobic? High intensity. It might be. We put them individually because we wanted to see individual performance. It was, by the way, we had a lot of critics from opponents because social isolation is also stressor. But we had to do it because otherwise we could not know which reach how much running. Okay, then I allowed my rats to run for 3 weeks. After 3 weeks, they were anesthetized, and I checked the activity of serotonergic neurons, and what I see. This is how it looks like an average serotonergic neurons in control rat. And when I started to measure serotonergic neurons in rats, in running rats, in rats which we allow it to run for 3 weeks, I could not recognize because I never saw serotonergic neurons of such a higher activity.
Intensity is also much higher. The upper one is 1 millivolt and the low one is 5.
Well, amplitude, in extracellular electrophysiology, amplitude is not a physical marker because it's not informative, because it also depends on how far you electrode from the neuron. And it just so we never look to amplitude in the extracellular recording. And here we see that we have more than 200% increase in the average spontaneous activity of serotonergic neurons in the running rats. But what with noradrenaline and dopamine neurons, we did not see the big difference. Then there was question how drugs, how physical exercise will interact with a classical pharmacotherapy treatment. This is you saw already this illustration, is an illustration taken from this study. This is a control sedentary rat. We apply escitalopram, we have desensity, we have downregulation, we have neuron firing activity is going down to 0. When we inject whey, then we have recovery of the neuron firing activity. And this is serotonergic neuron from running rat. We inject escitalopram and we don't see any effect, any inhibitory effect. And when we inject whey, we have even increase behind the puzzle level. So probably what's going on, probably voluntary physical exercise induce some kind of desensitization of early desensitization of autoreceptors. And co-administration of, if physical exercise definitely cannot be the main treatment in the severe depression. Yes, the microdialysis study which found increase in serotonergic concentrations in the several brain areas. And this is for the same study of QAM, the same meta-analysis study of QAM. He also found that if you combine of physical exercise with antidepressant with physical exercise, you get higher efficiency of the treatment than if the treatment alone. The only big problem when my postdoctoral supervisor Pierre Blier saw my paper, he told me, Eli, you are not a psychiatrist. You never saw depressed patients. No way that you will make them to exercise. Well, last question for today's lecture. If the sensitization of presynaptic receptor is sufficient for drug response or not? What you think about it? Let's go back to this. To this one, our first figures. No. Yes, this one. Okay, in 2 weeks we have the sensitization of receptors, of autoreceptor, and we start to see most first signs of the antidepressant response. But full response we see much later, few weeks after it. It means that this is probably a very important step. After this desensitization of the autoreceptors, you see some initial effect, but you don't see the full effect of the treatment yet. It means that some more factors must be involved not only desensitization before we see the effect of the antidepressant drug. And the reason to this situation is because depression is actually, we will come back by the end of the course to this very important point. Depression is actually disease of neuroplasticity. Decrease of neuroplasticity is the reason of depression, and the treatment of depression, successful treatment of depression is the restoration of the neuroplasticity. Neuroplasticity in the mammal in our brain is expressed in several levels. One level, I call it micro level, it's neurogenesis. Depression is strongly associated with neuroinflammation and loss of neurons, particularly in the hippocampus. And antidepressant treatments associate the increased neurogenesis. I believe everybody heard about adult neurogenesis. It was one of the most significant discoveries of the 20th century. And later it was confirmed that it's pretty strongly involved in pathophysiology and treatment of depression. And what the factors, inflammatory cytokines and stress hormones, such as glucocorticoids, they contribute to the neuronal apoptosis. And monoamines, the between of the factors which contribute to the creation, to the neurogenesis. But neurogenesis is only one level, micro level of the neuroplasticity. Neuroplasticity is also expressed, I call it the nano level and pico level. Nano level, it's a reorganization of the dendritic trees. And depression is associated with hippocampal and prefrontal cortical dendritic trees. They much less branchy, they much less dense in patient with chronic depression. And third level is a pico level. It's a level of dendritic spines. I believe you heard about dendritic spines. Depression is associated in the decrease in the density of dendritic spines. But it's only about cortex or hippocampus. If we speak about other brain areas, such as nucleus accumbens, which is other way around. Depression is associated with a more developed dendritic trees and higher density of the dendritic spines. And the molecule, which is the most fundamental molecule in the neuroplasticity, is the BDNF, brain-derived neurotrophic factor. It's a protein which involved in all levels of neuroplasticity, from neurogenesis to reorganization of the dendritic trees. And this shows that DNF can, what is very interesting in the BDNF signaling, BDNF can, this is a molecule which reinforces its own synthesis. Usually, classical biological mechanism is a negative feedback. In case of BDNF, we have positive feedback. BDNF activate tyrosine kinase B receptors, which stimulate expression of VEGF. VEGF is not abbreviation; it's a name of the molecule. I don't know why it was called this way. And VEGF, it's transcript of VEGF. It consists of several proteins. When C-terminal of this transcript get cut out, it's called TLQP62. And TLQP62 is reinforced expression of BDNF back. And their mechanism, as far as I know, still not known. And this is a short loop of positive reinforcement of BDNF expression. There is also a long loop, and this long loop involves serotonergic neurons. It is known that BDNF increases the excitability of, well, it's not yet known, I'm working on it now, but it's strong evidence that BDNF increase excitability of serotonergic neurons. And it's very well established, it's a work of Tumor, from the laboratory of Tumor, Ronald Newman, that 5-HT via probably 5HT1A autoreceptor increase and via cyclic IMP and cyclic responsive binding mechanism in a reinforced expression of BDNF. And it seems to be that, well, this is mechanism of effect of BDNF probably involve calcium signaling, and it's linked with extracellular and intracellular calcium channels. And it's very strongly involved in the antidepressant drug effect. This is figure from my review papers, and it shows how the effect of BDNF, how antidepressant drugs effect involve BDNF, and how involvement of BDNF leads to the remission of behavior effect. First, we see what happened after patient take antidepressant drug. We have increase in extracellular levels of 5-HT and activation of 5-HT autoreceptor. It happened within seconds. And it resulted in decreased excitability of 5-HT neurons like we saw today during our today class. After 2 weeks, we have desensitization of autoreceptor. We have restoration of the excitability of 5-HT neurons, and then we start to see the partial response. When we have restoration of the firing activity of neuron, we have increase in the 5-HT levels on the nerve terminal area, particularly in the hippocampus, and then we start to see increase in BDNF expression, which probably leads to this partial response. What is very interesting that BDNF expression can reinforce itself by mechanism involving activity of 5-HT neurons. Is my idea. I believe that in this state is very critical because some kind of equilibrium or steady state between BDNF expression and activity of the neuron must be formed before we go to the increase to the neuroplasticity and finally to the remission. I think by this I finished what I wanted to deliver about autoregulation of 5-HT neurotransmission. We can spend few minutes per question, and if time will be remained, we can start the next topic. Yes, question please now.
So from the side, because it means that if I can make the increase neuroplasticity, I can, without, like, if there's a different way to reduce neuroplasticity, this would be another neural.
I think so, and I'm working on it. It's one of the topics running in my lab, so maybe we can bypass all the steps.
Do you know of any research that connects depression or stress to oligodendrocyte or other brain-wide?
Definitely, it's become very hot topic in the last years. There are few papers. I don't remember them right now, but I know that my good friend and colleague from postdoc, Bruno Gouillard, working on it.
Because neuroplasticity is not just the neurons being affected.
Definitely.
Myelin, it's everything together. So,
You would like to do this as a final work? It will be an excellent topic. Some more questions? Yes.
I have a question on the side when you showed the growth of I don't understand the graph.
Which one?
Well,
Okay. What? This?
next one. Okay. Anyway, how do I understand the GNN?
Oh, this is not question to ask me.
Oh, wait. There's from this, if exercise is the best, how do you see it?
It's meta-analysis. I never did meta-analysis myself. It's meta-analysis when you took a result of the several studies and put together. Somebody did meta-analysis of you? Because psychologists, they frequently do meta-analysis.
It's because you have such a low number of participants, so you have to mash everything together.
On the rank exercise, how do you, like, what is equivalent? This result is not only for them to do this.
No, they're not forced.
Their own.
Yes, there's a voluntary physical exercise.
So, like, I want the equivalent is just, okay, everyday walk to do, or are we talking about real?
Well, in
We need to be forced to, even if you like.
Well, in humans, they usually is voluntary. If it's not in Tiranut or
That causes more depression, not less.
yes.
Yeah.
In rats, we have two models. We have model for voluntary, when you just, it's special cage with access to the running wheel. And there is also model for forced exercise, the treadmill. Treadmill like we use also in gyms, but rats, they get, if they late and not run, they get slight electrical shock to the tail, and
So does it try that? But the effect of the physical exercise is similar?
yes, it's very interesting. It's also a problem more like anxiolytic and antidepressant, like even forced exercise. But for this, we wanted to use voluntary model, because in voluntary model you can also make correlation to check how the more questions. Should we start next topic? Okay, we will. Okay, today till now, next topic which we will briefly start today in the time which is remaining, and it will be a topic for the next class mostly. It's interaction between different monoamines. Today we were very focused on serotonin, and we forget about for a while we neglected other two important guys, noradrenaline and dopamine. And we will talk about it. What we spoke today is that one of the problems with antidepressant drug response is a delayed effect. But delayed effect is only half of the problem. Another problem is a limited efficiency. Most of the antidepressant drugs, they either, at least when you take patient and you treat patient and give him her first choose an antidepressant drug, only in 30% of the cases you will see response, and 60% the patient will not respond to the first chosen antidepressant drug. Another problem is a partial response. You saw depressions have pretty strong link of the symptoms. In some cases, patients get less anxious after a while. You treat patient with escitalopram. After 2 weeks, that patient report, yes, I'm less anxious, but still have depressed mood, still have fatigue, and still have sleep disturbances, or other way around. And another problem is a relapse. Sometimes you treat patient, After several weeks, it's a good relapse, but after a few months, it's a symptom of depression, or some of they come back. What was found, what was very interesting, if you combine several antidepressants, and especially if you combine several antidepressants with a different mechanism of action. For example, you combine SSRIs with mirtazapine. SSRIs is a serotonergic drugs, and mirtazapine is a serotonergic and noradrenergic drug. Then you have better response, and then you have more chances to lead to reach long-lasting remission of depression symptoms. And then why it is so? It is so maybe because different symptoms, they linked to different neurotransmitter system. And if you use multimodal treatment, which touch several neurotransmitter system, you have a better chance to live remission. For example, SSRIs, the primary effect will be on the depressive thought. When people obsessively think about their worthlessness, and if you touch serotonin and noradrenaline together, you have better chance to improve anxiety. Noradrenaline, if you touch adrenaline, you can improve such a symptoms like such a fatigue. It's a chronic tiredness reported by them. And finally, anhedonia. If we speak about anhedonia, then dopamine can be probably the more promising targets to target. But what makes the picture much more complicated, because all these neurotransmitters, they have different roles, but they're very interconnected on the functional level. We remember from today's class that serotonergic neurons, they autoregulate themselves via 5-HT1A and 5-HT1B autoreceptor. Noradrenergic and dopaminergic neurons, they also autoregulate their own activity. Dopamine is D2 receptors, and noradrenaline is alpha-2 receptors. But in case of noradrenaline and dopamine, unlike serotonin, same receptor, as far as I know, same receptors, they are nerve terminal and on cell bodies. But all these neurons, they also regulate each other. For example, 5-HT. When you have increase in serotonergic neuronal firing activity, an increase in the serotonin release from nerve terminals, it leads to inhibition of noradrenaline neurons and dopamine neurons. Serotonin usually negatively regulate catecholamine neuronal firing activity. There are different receptors. In case of 5-HT1A of noradrenaline neurons, 5-HT2A receptors are involved. And in case of 5-HT of dopamine neurons, 5-HT2C receptors are involved. What is very interesting from an electrophysiological point of view, 5-HT2A and 5-HT2C receptors, they excitatory receptors. Usually this activation of the receptors leads to activation of the neuron. So how it can inhibit? And interneuron is involved, and it's a GABA interneuron involved in serotonin dopamine, and serotonin noradrenaline. I don't have pointer. I will put it back in this interaction. Finally, noradrenaline and also noradrenaline and dopamine, they also regulate each another. This is my study from postdoc. We show how serotonin dopamine interaction, serotonin noradrenaline interaction are working. What's happening if we treat animal with escitalopram? So after 2 days of treatment, after 2 days of administration of escitalopram, it leads to inhibition of noradrenaline neurons, same as what we found with serotonergic neurons. citalopram, which you remember is less efficient as a reuptake inhibitor. After 2 days, we don't have effect of noradrenergic neurons because citalopram is not efficient enough after 2 days to develop a sufficient increase in serotonergic concentration of near terminal level. And our findings with the citalopram were very surprising, and reviewers did not even believe that it's a really linked with serotonin. They told us maybe the citalopram still has some noradrenergic effect. And to answer reviewers, we added PCPA. PCPA, it's, don't ask me to spell it, but it's inhibitor of 5-HT synthesis. And inhibition, if you inhibit 5-HT synthesis, then citalopram does not work anymore, because if you don't have serotonin, you have nothing to block its reuptake. And this is serotonin-dopamine interaction, very similar picture. This is rats which treated 2 days with escitalopram. They have much less activity of dopaminergic neurons. And again, to prove that it's indeed mediated by serotonin and not any kind of direct effect of escitalopram on dopamine neurons, is my core study was done similar time. We were together in postdoc with Bruno Gouillard, who is now in Toulouse in France. He did something else. He make lesion of serotonergic of the raphe nucleus. He destroys the raphe nucleus, and rats without serotonin, they have much higher dopamine neuronal firing activity. This is method that I really don't like, because if you destroy serotonergic, somebody did it of you or so these rats, they enormously aggressive. Rats, if you destroy raphe nucleus, you must keep them separately because they will kill each other in the cage. And if you just open the cage, animal rat attack you. So the short term, yeah, the good question, what's happened after? Exactly. Thank you for the question. What do you think? But perhaps you can say perhaps we will see the same desensitization, and this problem will be solved. First, you see, it can be a problem, because indeed we need to increase serotonin to treat depression, but we increase serotonin and we inhibit dopamine, noradrenaline, neuronal firing activity, and it may be not so good in terms of concentration, of fatigue, and of anhedonia. But maybe you tell me this receptor desensitize, and after desensitization, you will see the restoration of the normal neuron firing activity. And let's see what's going on. No, unfortunately, unlike serotonin, it's not the case. Here is a 2-day treatment with escitalopram, exactly same level of inhibition you have after 14 days. If it will be serotonergic neurons, we will see total restoration of the firing activity. With dopamine, the same picture. With dopamine, we have inhibition, and after 14 days, we have restoration of the neuronal firing activity. We don't have any restoration, unlike with serotonin. It means that what we saw with the serotonin, we have restoration of the firing activity and increase in the neurotransmission. In noradrenaline and dopamine neurons, we don't have spontaneous recovery of the inhibited firing activity. And what can be done to solve the problem? One of the methods which used to solve the problem is to treat patients with atypical antipsychotic drugs. Co-administration of antidepressant with, we already spoke a little bit in previous class about antipsychotic drugs. We know that classical or typical antipsychotics, they act almost only on D2 dopamine receptor, but 5-HT2 areas, but atypical antipsychotic drugs, they act on plenty of other receptors, such as 2A, 5-HT2C. You already remember these guys, which are involved in serotonin, noradrenaline, and serotonin dopamine interaction, as well as other receptors. And this is future antipsychotic drugs. We will come to the triamine receptors and adenosine receptors in the next lectures. And this indeed what we found, that if we could administer escitalopram, this is escitalopram, this is vehicle. Escitalopram leads to inhibition of the noradrenaline neuronephric activity, but if you give together escitalopram with risperidone, a typical antipsychotic drugs which act on 5-HT2A and alpha-2 adrenergic receptors, we have not only restoration, we have even increased the nephric activity of noradrenaline neurons. But I think that today probably everybody already tired, and this topic is pretty complicated. So after this short introduction, with your permission, we will stop today. And with remaining time, I can take few more questions.